Hello, welcome to some more video notes. Today in Topics 2, we'll be covering Topic 2.5. We'll be doing the whole thing, and we'll be going through basically what enzymes are. So for quite a while, we've been talking about proteins and how proteins do various things inside of the cells. They do thousands of different roles in some regards. And so we're going to talk about the most common form of proteins, and which would be the enzymes, and how enzymes actually function to keep your cells alive by powering these kind of chemical reactions. And we'll also talk a little bit more about optimum temperatures, denaturization, optimum pHs, and then how we can use enzymes in industry in order to gain materials in a lab that we wouldn't necessarily be able to have access to without the use of these enzymes.
No, God! No, God, please, no! 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 No!
So we will be covering all of this content. So we'll be looking at these things, first talking more about active sites and substrate binding sites and how enzymes go through their activity. We'll be looking at the rates of activity of enzymes. We'll be looking at how temperature, pH, and substrate concentration can also affect those rates. Do a little bit of a review on what denaturing actually is, and then we'll be looking more at immobilized enzymes in industry and how we can use them to make lactose-free milk. All right, and then of course some of this other stuff will be kind of brought up as we go through the PPT. All right. So you need to make sure that you have a good understanding of what an enzyme actually is. So let's start with our definition. So you think of an enzyme, it is a globular protein. They're always globular. There are no fibrous enzymes. Only globular proteins are enzymes. And they are going to be biological catalysts. And so that basically means is that they are part of a living thing. All right, they are catalysts, kind of like different compounds, different materials can be catalysts in chemistry, but they're made from organic material. So they're biological material, and they are going to be involved in helping a biochemical reaction by lowering the activation energy, which basically means making it easier for that chemical reaction to occur at a specific temperature or around a specific level of energy. And so by making it possible for that chemical reaction to occur faster or happen more likely, it is allowing that chemical reaction to happen in order to keep the cell alive. So in order to get into all this stuff, we'll eventually be talking about some of the basics. Won't really be getting too much into inhibitors. That's our HL content, so don't worry about that if you're an SL student. And we'll be talking about allosteric sites and inhibition a little bit in the HL content as well. So really it's the basics of what we'll be focusing on in these notes. So if you're going to understand how an enzyme functions and you're going to correctly describe it, you need to be using some key terms like the idea of a substrate and an active site. And so this little diagram on the side is a really good way of understanding what it is that we're talking about. So a substrate, or the specific term substrate, refers to the reactants of a biochemical reaction. So remember, chemical reactions always have reactants being changed into products. Well, the reactant that starts our chemical reaction in terms of a biochemical reaction would be therefore called the substrate. And so the substrate should have a very specific enzyme that is designed to interact with that substrate. And so a globular enzyme will be able to catalyze the biochemical reaction related to that substrate. And the reason why they're connected to each other is because the substrate is able to fit into the active site of the enzyme. And so the enzyme will have a very particular physical shape. Right, so here you can see it's kind of shaped like that arrow-shaped substrate, and so because of it having that very specific shape, that active site, that substrate will fit into that space, and when it fits into that space, it will be able to bind to the enzyme, and then the enzyme will be able to go through the chemical reaction. So when this occurs, when the active site when the substrate fits into the active site and binds to it, then we create what we call an enzyme-substrate complex, or basically a fusing of the enzyme and the substrate together. Now, during the fusion of an enzyme-substrate complex, normally the enzyme doesn't have something inside of its active site. That's the normal state of the enzyme, is to be empty. So when a substrate comes in and kind of forces itself into that active site, it causes a slight conformational change, or basically a physical change of that enzyme. And so because of this physical change the substrate puts on the enzyme then also puts a little bit of a physical change or physical pressure onto the substrate, which helps drive the chemical reaction. And so by doing this, it lowers the activation energy or increases the probability of the chemical reaction to occur. And so in this instance, in the diagram, what happens here is that there's some type of physical reaction, or chemical reaction that allows this to separate. And so it breaks the substrate basically into two. And you should remember that that means this must be a catabolic enzyme, because catabolic things will break bonds, right? So we break these bonds, right, between these two parts of the substrate, and so then the substrate ends up becoming the products. And so two products are produced as a result of this chemical reaction. And so those two products, once they form, are then released from the enzyme. They leave the active site. And the thing is that we talk about it being a catalyst. In a chemical reaction, a catalyst can be used over and over and over again, and it never really disappears because it's taking part in the reaction, but it's neither a reactant or a product in the reaction. So it can just be recycled over and over again. And an enzyme is the same idea. So this enzyme here can be reused again. So another one of these substrates can end up inside the active site, and then the whole process can repeat. And so that's why, even though there are thousands and thousands of different enzymes inside of your cells don't necessarily need to have thousands and thousands of copies of the same enzyme, because we can use an enzyme multiple times, actually hundreds of thousands of times. An enzyme can just be doing its job, rather than having it, happen once and then we have to make a whole new enzyme. So this idea that we can recycle them in terms of the way they are a catalyst really helps speed up chemical reactions and makes this whole process more efficient. So another thing to remember is that because of the active site is a very particular shape that normally goes along with the substrate, enzymes are said to be relatively specific to their substrates. Now there can be instances where multiple substrates can fit into the same active site. So to give you an example, the same enzyme that you use in your liver to break down alcohol can also break down a toxin in antifreeze. So for some reason, if you got antifreeze, inside of your body, and it got broken down, and it would produce a toxic material that could kill you. However, if you did get antifreeze inside of your body, well, if there was alcohol in your blood, like if we had you drink pure alcohol, then your liver would focus on breaking down the alcohol, and it wouldn't focus on breaking down the antifreeze, because they both react with the same enzyme and the same active site. So the alcohol gets broken down, and then the antifreeze eventually gets removed from your body by your kidneys, and you're fine. You won't necessarily you'll be sick, but you won't actually die. So because substrates are very specific to their active sites of enzymes, we kind of summarize this as what we call a lock and key scenario. And so you've probably seen locks, and you know about keys, so a key has to fit just right into a lock in order for the key to open or close the lock and have the lock mechanics function. The same thing regard to the physical structures of our substrates. The substrates have to be just the right shape in order to actually fit inside the active site of the enzyme, which then will allow the enzyme to be able to go through the whole process of catalyzing the reaction. So in this instance, the substrate is the key and the enzyme is the lock. And so both structurally and chemically, they have to match each other in order for the whole enzyme process to occur. Now, even though the lock and key method was established early on in the discovery of enzymes and how enzymes and substrates interact with each other, as we do more research into how enzymes function inside the cell, we do realize that there actually are quite a lot of different substrates that can activate in the same, so there can be multiple, not a lot, but there can be multiple substrates that can fit into the same active site. And so those substrates, even though they might physically be similar to each other, they are not chemically similar to each other. Or they might be chemically similar to each other, but not so much physically similar to each other. And so to account for this, the more modern idea of how enzymes actually function are what we call the induced-fit model. And so the induced-fit model says that this active site here is kind of flexible. So the right substrate if the right substrate fits into this active site, the physical changing of the shape of the active site, here you can see it kind of got a little bent from its original structure, eventually leads to powering the chemical reaction, the catalyst for powering this chemical reaction. And then when the products leave the active site will then return to its original shape so that it can be reused again. So we said that it's an induced fit, so it's somewhat of a correct fit, but what happens is that because the active site is a little bit more flexible than we thought, if the right substrate enters the active site can kind of be flexible and move and form around the substrate and still allow the chemical reaction to occur. And so because of this stress that the active site puts on the substrate, that physical stress here you can see in this section here of our substrate in order to break them into two different products. That physical stress helps power this chemical reaction, which ends up lowering the activation energy of the chemical reaction. Now, just because a catalyst and a substrate are around each other doesn't necessarily mean that they're definitely going to have a chemical reaction. Substrates and enzymes are designed to interact and physically match each other, but that doesn't mean that an enzyme is attracted to a substrate. If an enzyme is around a substrate, it's not going to actively try to go over to the substrate and grab it, right? So there are other factors that are going to influence how efficient enzymes are going to function. So first off, substrates have to the molecule has to find the active site. And so for this to occur, they physically have to bump into each other. There has to be a collision where the substrate actually bumps into the enzyme, or vice versa. The enzyme has to bump into the substrate, whichever way you want to think about it. Not only does that have to happen, but the active site has to bump into the substrate in just the right way that the active site and the substrate are capable of binding to each other, right? So in order for this to happen, first off, one of the things we're talking about is globular proteins, which are able to dissolve in water. So then we'd also be talking about substrates that are also capable of dissolving into water. So this would have to mean that we're talking about particles dissolved in water, and so they're freely moving around in the watery solution, which allows them to, twist and turn and move in all different directions and eventually collide into each other. So when we're talking about enzymatic reactions, we're talking about all this occurring in an aquatic environment, in a watery solution. So we all know, or you might have heard before, this idea of Brownian motion. It's this idea that atoms don't stay still. Atoms are constantly vibrating. They're constantly moving. And so if the atoms inside of a substrate are moving, all of those combined vibrations eventually means that the substrate is also constantly moving. It's constantly vibrating a little bit. And so all of this random Brownian motion is driving random movement of the substrates and the enzymes in random directions. And so what happens is that eventually the enzymes are going to bump into a substrate because they'll have just the right angle, just the right force, and they'll fuse together, creating the enzyme substrate complex, which then allows the enzyme, the enzyme to catalyze the chemical reaction. Now, there are some things that we could do that might increase the probability of them interacting with each other. So for example, if the enzyme is immobilized, right, there's a definite surface of the membrane, for example, the cell's membrane. If it was stuck inside of the cell's membrane, well, there might be a greater chance that the substrate ends up bumping into that specific part of the membrane because of, concentration gradient differences between the inside and outside of the cell. If it's not immobilized, there can be a relatively large distance that these enzymes and these substrates might move away from each other. And so the distances between the substrates and the enzymes are going to be an influence about how quickly this enzymatic reaction is going to occur. The bigger the distance around them, the harder it is that they might actually bump into each other. This also has to do with size as well. The bigger the substrate or the bigger the enzyme, the slower that enzyme and substrate is going to move randomly. So that means they're going to be traveling less distances and they might bump into each other less often and with less force. So collisions are really going to be only a result of this random movement of both the substrate and the enzyme. And so we also have to make sure that those collisions are just at the right angle so the active site actually, binds to the substrate. The active site has to be the one that actually bumps into the substrate. So only through a successful collision do we actually get the enzymatic reaction occurring. So all of these factors have to be overcome in order for us to have a quick enzymatic reaction inside our cells or outside of our cells, right? All of these things are working against the chemical reaction occurring, because it's only going to happen if we get the substrate and the active site of the enzyme next to each other and at just the right force and the right angle to successfully have them fuse. Now, there are some factors that can help increase Brownian motion, and one of those would be temperature. Higher temperatures mean more energy is going into the molecules, into the atoms. So they start to move more, they vibrate more, which means that they travel greater distances because they have more random energy. So by increasing temperatures, we actually increase the probability that substrates and enzymes end up bumping into each other. So that's one of the reasons why your body temperature is as high as it is that having a higher body temperature increases Brownian motion, which increases the probability that your enzymes and the substrates in your body end up bumping into each other, which makes your cells a little bit more stable. However, it's also a little bit dangerous to increase temperature because increasing temperature can also lead to denaturization. So remember, we talked about this before. To denature an enzyme means it loses its three-dimensional shape. The tertiary structure is going to be broken. If the tertiary structure breaks, that means the active site is probably going to change shape. And if the active site changes enough, the enzyme will no longer function. It is going to be denatured, and so it will not do the chemical reaction. So we have to think about how things like temperature and pH as well, which we'll talk about, and then just the general concentration of substrate actually influence the rate of an enzymatic reaction. So real quick, just to give you a nice visualization of what I meant when I talked about denaturization. So again, if denaturization happens, then the active site is probably going to be changing shape. So here we have an enzyme before it is denatured, and here we have an enzyme after it's been denatured, before it's been denatured and the substrate can fit inside the active site. Here, after it's been denatured, the active site has been changed, and so because of that, the substrate can no longer fit inside of the active site. And if it cannot fit inside of the active site, the enzyme cannot perform the chemical reaction. So, though temperature can increase the rate of an enzymatic reaction, pH has an influence as well, and so does substrate concentration. So you need to know about these three key factors, what they would look like in terms of the overall effect on rate, and graphically what we would expect to see happen if we continued to increase temperature, pH, and substrate concentration. What is it going to do to the overall enzymatic rate? So I will go through each of these in the next few slides. So first, let's look at the idea of temperature. And so here, looking at temperature in degrees Celsius, we can see our enzymatic rate here is shown in this blue line. And so as we increase temperature, we start to see a significant increase in the rate of our enzymatic rate. And then once we hit a certain point, it doesn't matter how much we increase the temperature, we start to decrease significantly the enzymatic rate. And so here, what they're saying at this point, this is our optimum temperature. Our optimum temperature, here they're saying about 40 degrees, which is true for most enzymes in the human body. Most enzymes in the human body, really once you go above 40 degrees, they no longer function. They start to denature. And so the activity is increasing because the enzymes and the substrates are moving around each other faster, they're colliding more often, and so the chances of the chemical reaction occurring are happening more. However, once we go above the optimum temperature, we start to denature the proteins, and when we denature those enzymes, they no longer function very well. So here, at lower temperatures, obviously, the rate is going to be a lot slower because there's less kinetic energy, there's less random movement. So the enzyme can still happen, it can still do its job, but it's going to be really, really random whether or not it actually gets an opportunity to bump in the substrate. Things are moving slower, but then as we increase temperature, they start moving faster. So we have higher enzymatic rate because there's more energy, more kinetic energy, which means more frequent collisions, eventually hitting our optimum temperature, or what we would call our peak temperature. Then, once we go past the peak, we decrease our rate because we are now disrupting our hydrogen bonds, and now we are denaturing or losing the effectiveness of our enzyme because it just can't function anymore because the temperatures have destroyed the tertiary structure. So now let's look at the effect of pH. The interesting thing about pH is that pH in general is always going to follow the same path, where we have a significant increase in our activity, we hit our optimum pH, and then once we go above our optimum pH, we see a significant decrease in activity. However, the thing that makes it interesting is that different enzymes are designed to have different optimum pHs. So for example, here we have pepsin. Pepsin is an enzyme that's in your stomach. It's in your stomach acid, and it's designed to help you digest proteins that you eat, for dinner. However, its optimum pH is around 2, which is really, really low compared to most other enzymes in your body. Most enzymes, for example, here we have salivary amylase, which is the amylase in your saliva, basically. Amylase, if you remember, is the enzyme that's responsible for digesting starch, right? And so in your mouth, you have a pH of about neutral, right, about 7. And so the optimum pH for amylase is about 7. So we don't need enzymes that have an optimum pH of 2 in your mouth or in other parts of your body, because you don't have pHs of about 2 anywhere in your body besides your stomach. So enzymes that are going to function in your stomach need to be designed so that they function really, really well in that pH. Enzymes that are going to be in different parts of your body, whether it's something that's acidic or basic or more alkaline, they're going to have to be fitted to work in that environment so that they are somewhere near their optimum pH. Now, there can be fluctuations in the pH inside of your mouth and inside of your stomach, but they should in general function best in either a neutral, acidic, or basic environment. And if you take them out of that environment, their rates are going to decrease to the point where they might not function at all. And you can even denature an enzyme if you put it in too acidic or too basic of an environment, because then again, it will disrupt the bonds inside of the tertiary structure, causing it to lose its active site and not be able to function anymore. And so the last one we're going to look at here is our substrate concentration. And so as we increase substrate concentration, as we have more and more substrate around our enzymes, we expect our enzyme activity to increase, right? But it's only going to increase to a certain point. It's going to hit what we call its maximum rate. And so see here, the rate is going up, it's going up, it's going up, it's going up, and then at a certain point, we say right here, whatever this concentration is, it doesn't matter if we have more substrate in the solution, because essentially at this point, all of the active sites have been taken. So originally, by increasing the substrate, you're increasing the chance that there's a collision with the active site, and therefore we have a faster chemical reaction. However, at the optimum concentration, all of the active sites are being used. And so since the maximum number of active sites are already being taken, and any extra amounts of substrate added to the solution isn't going to make the rate go any faster, because it doesn't matter if more and more substrate is present, there's only so many active sites that are there in order for the product to be created, for the chemical reaction to be built. So I want you to imagine that, I have 3 students, and I want them to build me a really simple Lego house, right? So if I put a few Legos in front of them, and I tell them each of them to build me a house, all right, because they're randomly picking up these Lego pieces, it's probably going to decrease. They're not really going to work too quickly through this process of making the house. If I give them each a lot of Lego pieces, they're increasing the probability that they grab the right Lego piece, and so they're able to make that house faster than if they didn't have too many Legos. However, if I give them a room full of Legos each, well, now it really doesn't matter that they have that many Legos, because it's really the limit is how fast they can even build the Lego house. So that's kind of where this idea comes in with our substrate concentration. The limit there is that there's not enough enzyme active site in order to keep doing a faster chemical rate. And it's really just a limit on how fast the chemical reaction can occur that's stopping the rate from increasing. So once we eventually hit our optimum concentration, the rate of the enzyme just won't go up anymore. So for this last section, we just want to talk about how are we going to be using enzymes in industry. And the thing is that enzymes are a major, major part of industry, because enzymes, like we said, are biological catalysts. They are these little microscopic, nanoscopic machines that can do very specific chemical reactions hundreds of thousands and millions of times over and over and over again, and they don't really get worn down, and all you have to do is give them the right environment and give them the substrate, and they'll do one really specific task. So we can use enzymes to do a lot of things in industry for us, rather than, depending on a whole living thing like a whole cell. So for example, we can use detergents that have proteases and lipases inside of them. If you've got stains, containing proteins and fats, well, if the protein and the fats are broken down to make them more soluble in water, it will be easier to remove them from the clothing that they've stained. So if we use a detergent that has enzymes like proteases and lipases inside of it, we could be more effective at removing stains from clothing. We can use enzymes to break down waste material like pieces of starch and grain that we're not going to be using as a food source, and we can use it to break it down into biofuels, because the conversion of material through anaerobic respiration, you remember, we can break down a starch and turn it into ethanol, and we can burn that ethanol as a source of fuel, right? So we can use enzymes to do that. We can make textiles, right? We can clean things, we can polish things, we can add certain features to things using enzymes. In brewing, right, brewing wine or beer or other types of alcohol, you're going to need enzymes to help along with the process. So using enzymes to go through the process of alcoholic fermentation is where we can then produce these types of products. Medicine. Right? Enzymes can be used in certain types of medical application. Enzymes can be used to make proteins that then can be used to form medicine. We can also use enzymes for altering DNA and creating genetically modified organisms. We use lots of enzymes in the food industry, right? If we want to get more juice out of fruits, we can use pectinase. Pectinase can help break down pectin, which kind of prevents the juice from really getting out of the cell walls of the plant cells. So if we use pectinase to break down the pectin that stops the juice from getting out, we can get more juice out of our fruits when we go through the process of making a fruit juice. So we can produce more juice faster. We could break down fructose. Right, and to get more, not fructose, we can break down sucrose into fructose and glucose. And by breaking that disaccharide down into monosaccharides, we can get like a sweeter taste, and we can modify the sweetness of our foods using enzymes. And we can even, produce cheese and other things through the process of using these enzymes. So there's so many different things that we could be doing, and also paper as well. Sorry, one more thing, paper. Breaking down materials and fibers in order to make different paper. Enzymes would be a good way to do this process over and over again very quickly, so that we can mass produce paper. So regardless of how we're going to be using the enzymes, there's one major thing we have to consider when thinking about how those enzymes are going to be utilized in this process. And it's whether or not is a free enzyme floating around in a solution, or is it an immobilized enzyme, like something that's stuck to a plastic sheet, or in some type of like gel aggregate bead or something that physically can be moved around. The problem is that if the enzymes are in solution, it's impossible to remove them from the solution. Remember, these are nanoscopic things. We can't really filter them out. We can't grab them or take them and get them out of the solution. So once they get added to something, they're just going to permanently be part of that solution, and there's really not much you can do about it. However, if we use something like immobilized enzymes, we could take enzymes and we could stick them onto something. For example, maybe we have a thin plastic sheet, and then we use special chemical reactions to take those enzymes and basically stick them to this sheet. And so if I want them to react with a solution, right, I can put that sheet into the solution, right, and it does all that chemistry, breaks all the material down, reacts with all the substrates. But then when I want to take the enzymes away, I can just remove the sheet that they're attached to. And so now all the enzymes are gone, and whatever this material is, it's now free of any enzymes. So doing something like that could really speed up the process. For one thing, it makes it so that we don't have to worry about removing or denaturing or getting rid of any enzymes in our solutions when we're done using them. The other thing is that we can use them over and over and over again, right? So if I can keep the enzymes, and if I can take them back out of the solution, then I can put them in a new cup and do the whole process over and over again. So here's some information to think about why immobilized enzymes are good. So first off, we can dramatically increase the rate of reaction, right? Because we can have real control over the concentration of the substrates, and we can also have a better increase the influence of the enzymes being able to collide with the substrate. So we could have basically more surface area where these enzymes are able to interact with the substrate. we can recycle these enzymes. We can, again, use them many, many times. If we can take them back, then there's no reason why we can't just go and use them again in another reaction. We can separate them from our product, which is great, because then, again, going back to the recycling thing, they don't become this waste that ends up inside of our product. We end up recycling them back in. So this separation, not only does it make the enzymes able to be used again, but also makes the product better, because now it doesn't have a bunch of enzymes in it. And maybe one of the best reasons is stability. So because the enzymes are stuck to something, they're actually a little bit sturdier, so they're less likely to end up denaturing in high temperatures or an acidic pH. So taking all these factors into consideration, mobilized enzymes are just a really good way to increase efficiency when we go through the process of using enzymes in some type of biological reaction, in some type of mass production and industry. And so to give you a little bit more context, we're going to go into a really specific example of how immobilized enzymes can be used to make lactose-free milk. So lactose intolerance, or this idea of not being able to digest lactose, is a medical condition that lots of people suffer all over the world. It's a little bit more common in more developed countries, and European and American and Australian countries. But what happens with lactose intolerance is this idea that you lack lactase, or you're missing the enzyme necessary in order to really digest lactose. And so instead of breaking it down into galactose and glucose, the lactose kind of builds up in your digestive system, and it can cause some problems in your digestive tract, maybe make cramping, sometimes can release some gases, and just general unpleasantness. And so someone who is severely lactose intolerant, they will not be able to enjoy, cheeses and milk and ice cream and other things built around dairy, because the lactose would make them very, very sick. So, move to that. So the main goal from lactose-free milk is this idea of using the enzyme lactase to basically process the milk so that all of the lactose is already broken down, so that you don't have to worry about getting any lactose in the milk, so that you're free of the lactose, and therefore any milk, cheese, or dairy that is made from this process would be free of the lactose and would be able to be consumed by everyone. So basically what happens is we get a bunch of lactase from a simple microscopic organism like yeast, for example, or other type of bacteria, and we get a high concentration of lactase. And then what we're going to do is we're going to use a special chemical reaction that creates what we call an alginated bead, or basically kind of a jelly bead. And so that jelly material that the bead is going to be made of is going to also contain just millions and millions of lactase. And so the lactase is going to be bound to these beads. And so then what we do is we can put them in a container like the diagram shows here, and we just kind of pour the milk through that container. And as it slowly moves through the beads, the enzymes on the surface of those beads are going to find any lactose that's in the milk, and it's going to break it down so that the lactose is no longer part of the milk. Now this process isn't going to be super efficient the first time. It's going to take multiple passes through a chamber like this. It's going to take, some slow movement through these beads in order for all the lactose to be broken down. But eventually, after redoing this over and over again, you will produce milk at the bottom that does not contain lactose. And so then that lactose-free milk is going to be mostly comprised of just glucose and galactose. So it will taste a little bit sweeter, because glucose and galactose, being simple saccharides, taste sweeter to us than something like a disaccharide. But I won't have to worry about any lactose being present. And the other thing, because these are immobilized enzymes, we can do this at some higher temperatures so that we can kind of speed up this whole process a little bit faster. And we can remove those enzymes by just, removing the beads from the container, or having the milk pass through and being collected at the bottom. So we don't have to worry about any of the lactase enzyme just sitting around inside of the milk. So if we wanted to increase the sweetness of the milk, for example, we could do this, if we want to make sweeter, nicer cream for, ice cream, for example. This would be a good way to do it. It also reduces the crystallization of ice cream, for example, because galactose and glucose are easy to dissolve into material. They're more soluble because they're smaller than something like lactose, and so less likely that crystals form when the ice cream goes through the freezing process. And then this can also be increasing the production time, shortening the production time or increasing the rate of the production of yogurts and cheese, because then we can take the bacteria and it can grow using the galactose and the glucose sugars rather than breaking down the lactose, which would take more time for the bacteria to digest and therefore would slow the bacterial's growth as we're going through the process of producing yogurts and cheese. So now that we've talked about how enzymes can be used in industry, the last little bit that we want to go through at the end of these notes is just how can we be using enzymes in laboratory experiments in general. So you eventually will be doing some laboratory experiments using enzymes in December as a kind of a practice IA. And then you also have your actual IA that you'll need to decide what topic it is that you want to research. And something you could do is you could design a lab around investigating some of the environmental impacts on, the rates of an enzyme or how well an enzyme functions at a specific reaction. So what would be the appropriate way to set up an experiment like that? So we could give an example of catalase. Catalase is a very widespread enzyme. It appears in lots of organisms because it helps deal with hydrogen peroxide, which is a very, very toxic byproduct of our general metabolism. And it can take hydrogen peroxide and it can convert it into just water and oxygen, which are two things that most living things are able to use readily, to survive. So that's a pretty good exchange, getting rid of a toxin and producing some water and oxygen. So if we wanted to research catalase, we'd have to think about what is the point of the investigation. What is the independent variable, right? What is it that we're trying to determine about something with catalase? Is it something with the environmental conditions of temperature, of substrate concentration, of pH? Would it be comparing different types of catalase using the same type of hydrogen peroxide? Would it be different types of cells and whether or not the catalase present inside of them is being as effective based on their origins? So these are all things we'd want to be considering. We could also look at the concentration of the enzyme itself. Obviously, a higher concentration of catalase enzyme would go through the reactions much faster than a lower concentration. Things that we'd want to consider, though, variables that we'd want to control. So obviously, we'd want to be controlling certain masses of materials or substrates that have been dissolved into our volume, or we'd want to have a consistent concentration of those substrates. We might want to keep, if we're going to modify the substrate concentration, but we want to still measure the rates, we'd want to think about how to keep the enzyme concentration the same. What are the exact units that would be, and what would be an appropriate range? If we're going to be looking at temperature, for example, if the optimum catalase temperature for humans is 37 degrees, does it make sense to be looking at 10 degrees, 20 degrees, 40 degrees, 50 degrees, or should it be a smaller range of increments, maybe 37, 35, 33, 31, in order to look at the effects of temperature on the rate of the enzyme. If it's too small of a range, it could be quite difficult to see differences in the results, because remember, you're not going to get perfect results, and so the variation within them might make a conclusion on your hypothesis difficult or impossible. If it's too large of a range, it might not really tell you anything more than you already knew, and so you're not really learning anything through the investigation. And then we also have to think about how we can safely handle these materials. So we already talked about the idea that hydrogen peroxide is dangerous because it is toxic. So you have to be careful about the concentration of hydrogen peroxide and how it is handled and how it is disposed of once the lab is done. But the enzyme as well. Biological catalysts like enzymes being dumped into an environment can have a negative effect on that environment. So we'd have to make sure that we had proper disposal processes. Maybe we need to boil the leftover enzyme to make sure that it's denatured so it's no longer active, or maybe we just need to dilute it, to a certain concentration before it can be, dumped down the drain and then be added to the aquatic environment where the drains meet. So these are all things that we'd have to consider when designing our experiment. Now, once we figured out how we want to model our independent variable and what controlled variables we might want to be looking at, we also have to think about our dependent variable. So if we're going to be measuring the rate at which an enzyme is doing some type of activity, what is the appropriate way to measure such a rate, right? And so really, there's only two ways that you can directly measure information about an enzymatic reaction. It's either measuring how quickly products are appearing or measuring how quickly the reactants are disappearing. So it's the rate that the reactants are being used, or it's the rate that the products are being produced. That would really just show over a specific time period, would show you the exact rate of the enzyme. But are you going to be doing this directly, or are you going to be doing this indirectly, right? Can you actually measure the amount of product that's being produced, right? If we're talking about catalase produces water and oxygen gas. Measuring the volume of water as it changes would be quite difficult because it's maybe an insignificant change. However, we could possibly measure the amount of oxygen gas that's produced using a chamber that can collect the oxygen gas as it's made and show displacement of water to give us a volume. Or maybe we just, count the number of bubbles over a certain amount of time that's produced from the reaction, and a way of counting the volume of the number of bubbles would be a way of interpreting the overall rate of the reaction. Could it be done indirectly, right? Since we know hydrogen peroxide can have an acidic effect on a solution, if we're kind of neutralizing that hydrogen peroxide, we could see that there would be a change of pH. So we're not really directly measuring hydrogen peroxide disappearing, but we are knowing that it must be disappearing because the pH is changing of our overall solution. So that would be an indirect way of measuring it. What type of equipment would be specific enough in order to make good measurements? If we're talking about volumes, we need to make sure we're using accurate equipment for measuring volumes. If we're going to be measuring pH, we need a relatively sensitive pH meter. If we're going to be measuring the amount of gas that's produced, we need a chamber that can be designed for capturing these gas and giving us a volume or quantity produced there. And we have to think about our think about the uncertainty of that equipment. So uncertainty is always plus or minus 1/2 of the precision. So the more precise a piece of equipment is, the more certain we can be, or the less uncertain we can be about our results. And we also have to think about how consistent are we going to be getting a readout from this equipment. We have to think about a time period as well. Are we going to measure the rate over just 30 seconds? Or are we going to measure it over like 30 minutes, right? That's a huge difference in time. 30 seconds might be too short to really see an overall effect of our independent variable. 30 minutes could be so long that other things start to influence our results. And of course, it would make the experiment extremely long to do 5 trials of our variants if we're doing 30 minutes for every trial. So we have to think about what is reasonable. And then we also have to remember the idea of doing things repeatedly so that we can calculate averages and make our data a little bit more reliable. With the IAs, the rule is that you should be doing a 5x5. That means you should have 5 variants. So if you're doing pH, you should be using 5 different pHs. If you're using temperature, you'd use 5 different temperatures. And you should be using 5 trials for each of those. So that means the experiment that you're doing, at a minimum, you're doing it 25 times. So that gives you a good amount of reliable data, but also is going to consume a lot of your time. And so you have to think about how are you going to space out this work so that you can fit it in, the time that you have to do your IAs. So getting into a little bit more about our controlled variables, we've done our independent and our dependent. Some things about the overall experiment that need to be controlled regardless of the other type of variables that have been selected. So obviously everything that's not our independent variable has to be controlled. If you're going to do pH, then temperature and substrate concentration, volume of enzyme, concentration of enzyme all have to be the same. Right? You can't change them because that makes your results unreliable. If you think about why we would use variable, or why these things, if they're not controlled, would affect your results. And so you should be able to comment on the reason for controlling them. So not just how you're controlling them, but why it's necessary to control them. You have to think about the exact procedure of how you're going to control them. What steps will you take in the method to make sure that they are the same? All right, if the controlled variable is too far from like an optimum, right, it could limit the overall experiment. So you need to think about like, well, what is a reasonable controlled variable? For that, it might be based on content that's been covered in class, but a good amount of it is really going to come from research. You're going to have to do a lot of research for a background section on the idea of the material. Right, and so what is it this enzyme does? What would be a reasonable control level for temperature or for pH based on the enzyme you're using? If you have an optimum temperature of 25 degrees for that enzyme and you're going to test pH, then you should probably be doing the experiment about 25 degrees. You don't want to be doing it like 40 degrees or 10 degrees because those temperatures are beyond what would be a reasonable level in terms of temperature. And so now temperature has become a factor even though you kept it at a consistent level. And then you have to think about what variables simply cannot be controlled and would therefore influence your results, and you have to maybe discuss them later in your evaluation about what those potential uncontrolled results did. And then last, we think about our safety, ethical, and environmental things. So I kind of commented this already, but it's always a need to make sure that we consider the health and safety of the people that are doing the lab and that you are doing the lab. Need to make sure that we're not injuring anything in the process of doing the experiment. And when we dispose of this equipment, we do it in a humane and safe way that it doesn't have a negative effect on the environment. Okay, so I know this would have been a really long topic. Sorry about that, but that is it for 2.5.